This is How Often, which is a project that we kind of worked on all together last year at the symposium. Well, this is one of the projects. So incidence rate calculation is one of the most common analyses in pharmacoepidemiology, whether it is used for comparative background rates for drug adverse events, assessing the potential impact of adverse events, or designing clinical trials. Here I just have the definition, and it's defined as the ratio of the number of cases to the total time at risk for patients in the target cohort. So why incidence rate? Well, first off, calculating incidence rates is relatively simple and it has fewer assumptions. And while incidence rates are more used for characterization and rates themselves are not meant to be interpreted from a causal perspective, it can still be really useful. So here I just have an example. If we know that the incidence rates are low and the side effect is not serious, then we probably don't need to worry so much about that. But on the other hand, if the incidence rate is high, then we probably still need to look out for that outcome, whether or not it's caused by the drug itself. So I said it's simple, but it still can be relatively complex. Previous OHDSI works have demonstrated that different factors like age, anchoring, database, and biological sex can influence incidence rate up to 1,000-fold. So we know that incidence rate estimates are sensitive to factors like age, sex, calendar time, and database. So the next question that we had was, does indication matter when it comes to estimating incidence rates? Well, we kind of do know the answer. And a really obvious example is if someone is taking a beta blocker because of a previous heart attack, then of course their incidence rate for heart attack after taking the beta blocker for that specific population is also going to just be higher. Now, what we don't know is what the extent of that variation is when we measure incidence rates by different indications. In addition, we're also interested in the relative contribution of drug indication to heterogeneity in these incidence rate estimates, and we want to compare that relative contribution to age, biological sex, and database. So, as I mentioned, How Often is our effort to characterize incidence of outcomes following drug exposure. And this was kind of started as a community effort last year. So we had different community calls in June and August to detail different aspects of this effort. We put out a call to the OHDSI community to get different research questions and develop phenotypes and draft a protocol. And if you were here last year at the symposium, we actually ran a full day How Often workshop. We had all of our data partners execute the How Often analysis package. We had lots of good discussions. And this is just one of many projects that resulted from the symposium last year. So going into more detail about the methods of our specific project, which looked at indications, we conducted the analysis on 13 different databases in October 2023, and obviously this was done last year during the symposium, as I mentioned. For the study design, the target cohorts are based on the first occurrence of a drug exposure in a patient's longitudinal record. We have 12 different drug classes, and each drug class was also further separated into different indications. Our outcome cohorts were 73 different outcomes, and they were developed by the OHDSI community, and they can all actually be found in the phenotype library. And I'll give examples of both the target and outcome cohorts in a second. Our time at risk is 1 day to a year after the cohort start. We stratified by age and gender, and for each of these outcome pairs, we calculated the incidence rate. And this is the 12 different drug classes. There is a typo on here. It's supposed to say GLP-1 agonists, but 8 of these drug classes had 2 or more indications, and 6 of them had exactly 2 indications. Beta blockers had 3 indications, and then TNF alpha inhibitors had 5 different indications. And then here's like a little sampling of the outcomes that we looked at. As I said, there's 73 total, so I just kind of pick and chose a couple from 3 different systems, but this is not an exhaustive list by any means. And then for analysis, we conducted a random effect meta-analysis of incidence rates across 13 different databases. We compared the difference in indications with a random effects model, and then we used variance components to quantify the relative heterogeneity between age, sex, database, and drug indication. And all of this was done in R using the Metaphor package. For our results, we had a total of 77,000 incidence rates that were calculated. And as I mentioned, 8 different drug classes had at least 2 indications. And then here on the rightmost column, you'll see the median variance component for each drug class. What this means is that for each drug outcome pair in that class, we calculated a variance component, and this is the median across all 73 different outcomes. And what this number tells us is how much heterogeneity can be attributed to the indication differences. In other words, if a drug class has high median variance component, the heterogeneity that can be attributed to indications is higher. And here we see that the drug class with the highest variation by indication is trimethoprim, and the drug class with the lowest variation by indication is GLP-1 agonists. And how do the variance components for indication compare to variance components for age, database, and sex? Here we show that usually either database or age have the highest median variance component, followed usually by indication, then biological sex. And the fact that biological sex usually had the smallest variance components is consistent with our previous studies that gender matters a little bit less when it comes to these variations that we see in incidence rate estimations. So some of the key takeaway and next steps. So among the drug classes that we looked at, trimethoprim is the drug class that is most sensitive to indication, and GLP-1s is the drug class that is least sensitive. And then for our variance components analysis, we found that indications contribute less to heterogeneity than database and age, but more so than biological sex. And we've actually been talking about this for a while, but the dream, but most importantly, my advisor, George Hripczak's dream, is to be able to scale up these incidence rate calculations to include all drugs and all outcomes. So you'll definitely be hearing from me and the team about how we can all work together to make this dream happen in the future. And my poster will be 120. Thank you.